Start recording. Okay. What first law of thermodynamics is conveying us? Energy can be transferred and transformed, but it cannot be created and or destroyed. So one form to another form, it can be transformed and transferred, but not created or destroyed. For example, plant can transform light energy into chemical energy. Okay. And then deer or animals may eat the plant and transform the chemical energy into kinetic energy. When you are performing the work, that is a kinetic energy. So everything energy is coming from this basically from this sunlight. What's the second law of thermodynamics saying us? During energy transfer, some energy becomes unavailable to do the work. So this means the some of the energy will be get wasted, usually as a heat. Logically, such loss makes the universe more disordered. Energy transfer or transformation increases the entropy of the universe. So whatever the heat energy releasing in every cell, every metabolic pathway, every animal or a plant, so is releasing into the universe that actually increasing the entropy of the universe. Entropy is a measure of systems disorder. It allows for the understanding of why a process will occur spontaneously. Entropy always increase, but because spontaneous system will always move towards the disordered rather than order. Entropy and spontaneity relation is what? The process will be spontaneous if it involves increasing the disorder. That means increasing the entropy. And it will be not spontaneous if it involves decreasing the disorder or increasing the order. So all anabolic pathways, what you understand, anabolic pathways mean synthesizing. Take a carbon, take hydrogen, take oxygen, or sulfur, all these things put into macromolecules like amino acids or carbohydrates. This is what? It is increasing the ordering, right? So it's not disordered, it's making more orderedness. So thus it is anabolic pathways are not spontaneous. That required a lot of energy to synthesize. And catabolic pathways, some of the steps are spontaneous and it releases the energy. And thus it releases the heat and also thus it will also increase the entropy of the universe. An increase in order will only occur if the energy is added to the system. Second law of thermodynamics alternatively can say for a process to occur spontaneously, it must increase the entropy of the universe. So that is another way of explaining or conveying the message of second law of thermodynamics. So free energy concept. Free energy is a portion of the system's energy that can perform work when temperature and pressure are constant. You can't estimate the amount of free energy itself, but you can estimate the change of free energy using this formula. So change of energy, free energy, change in free energy will represent change in enthalpy minus change in entropy or disorderedness of the universe. So we'll not go into detail of this thermodynamics. So then we move to the first metabolic pathway of the carbohydrate metabolism, glycolysis. So when we just talk about the energy and how the spontaneity of the energy related to the you know spontaneity of the reaction related to the disorderedness or energy, then we immediately move to the glycolysis where we get energy first. So this is a very important pathway. It is a must have in almost all living organisms. And if you observe another interesting thing, from the bacteria, lowest organism, until the complex organisms like human, all For all the cache energy is ATP. ATP is the molecule which gives the energy, which is, I mean, very surprising, isn't it? And and one more interesting thing is this glycolysis almost identical all the reactions from the bacteria until the human, but slightly some of the things could be vary, but the main concept of the each reaction is the same. And the catalyzing the enzyme, the each reaction, the 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 reaction and the catalysis is the same, but the molecules are different. Their structures are slightly different, but almost the same. So thus, we do share from bacteria until the human. We do share some of the genes which are needed for the fundamental uh, basic metabolic pathways. We still share some of the genes with we share with the E. coli or bacteria. So what is this glycolysis? This is a set of reaction that are oxidized glucose to pyruvate under aerobic or to lactate under anaerobic. So I think you might already studied that aerobic and anaerobic, and this glucose when it undergo oxidation under the aerobic condition, so it will convert into pyruvate at the end of the glycolysis pathway. Then, or in anaerobic conditions, it will convert into lactate. Both will be happening in our body too, even though um, we are consuming oxygen, we are aerobic, but sometimes in our body it will undergo anaerobic reactions to produce lactate also. We will try to understand those at the end of this glycolysis pathway, where it is required and when it will perform the anaerobic reactions. So where do it this glycolysis occurs in our body? So this glycolysis, as I we just understood, it is very basic uh, pathway which is needed for almost all the cells. So thus all cells of our body performs this glycolysis to get the energy, ATP, all the cells. But the excessive, more glycolysis will comparatively will occur in liver and yeah, extrahepatic mean other than liver, both. Okay, both will occur, but more glycolysis you can see in liver, and also you can see in all extrahepatic tissue. On cellular level, where it does it occur? It occurs in cytosol. So try to remember, try to visualize what is going on. So inside the cell, so in the cytosol, the glycolysis occurring, and different metabolic pathways occurs at different subcellular locations, different locations. So glycolysis occurs in cytosol. That means all the reactions, the enzymes, catalyzing enzymes are present in cytosol. So this is the summary reaction of the glycolysis. What is going on? Glucose converts into 2-pyruvate. So how many carbon atoms are present in glucose? How many carbon atoms are present in glucose?
6 carbons, sir.
6 carbons, very good. So 6 carbons are present in glucose. And pyruvate, can you guess how many could be?
3 carbons.
Very good. So 3 carbons. So six atoms containing glucose is splitting down to three containing carbons containing pyruvate to pyruvate molecules. So during this conversion, as just discussed, it occurs in 10 different steps, and it releases some of the free energy which is processing by the glucose in step by step until the pyruvate. That free energy in some of the steps, not all the steps, some of the steps captured into to convert in NAD into NADH, two molecules of NAD to two molecules of NADH, and two ADP with the two inorganic phosphate together forms two ATP. So some of the energy captured in the form of two NADPH and two ATP, and some energy is released as a heat. Then apart from that, it produces two water molecules and four protons. So this is the ultimate summary of the reaction of glycolysis. This is also called as Embden-Meyerhof pathway. Essentially, all cells carry out glycolysis. This is 10 reactions it contains, similar in most cells, but rates differ. So the pathway rate may be different, but almost all the reactions are same in all the cells. So in order to understand this whole glycolysis pathway, all these 10 reactions, so we can we can understand in, in a subsections of this, that is two phases we can understand. First phase is glucose into 2 glyceraldehyde 3 phosphate, where the one molecule of glucose is splitting down into two molecules. That means glyceraldehyde 3 phosphate also contains 3 carbon atoms, and glucose contains 6 carbon atoms. So first phase we can understand the pathway as converting the glucose into 2 glyceraldehyde 3 phosphates. And second phase, we can understand each glyceraldehyde 3 phosphate, how it become into pyruvate. That means 2 glyceraldehyde 3 phosphates, how it converted into 2 pyruvates, we can understand. What are the end products it will get? Products are pyruvate, ATP, and NADH. This NADH which produced inside the cell in the cytoplasm must be recycled. So we will understand actually why it need to be recycled uh, later. Okay, this is the whole overview of your carbohydrate metabolism. So one glucose molecules, which is containing 6 carbons, is producing 2 pyruvate or pyruvic acids by the end of glycolysis under aerobic conditions. And during this time, it produces 2 ATPs and 2 NADH. And ATP, which is instantly can be utilized by the cell or the body for the any kinetic reactions for the energy, any work can be done by this ATP. So instant cash, it got it. Then this pyruvate then and need to be converted into acetyl coenzyme A, which will enter into the Krebs cycle. So another pathway of the carbohydrate metabolism. So this step is another important step we can call is the second part of this carbohydrate metabolism, that is formation of acetyl coenzyme A from pyruvate, 2 molecules of pyruvate to 2 molecules of acetyl CoA. So this will also produces the energy, but in the form of only NADH. There is no ATP produced here, only NADH. And 2 carbons are released here in the form of 2 carbon dioxides. So thus, pyruvic acid is 3 carbon containing molecule and acetyl CoA is 2 carbon containing molecule. So now these 3 become now 2. So 1 is released as a carbon dioxide. So 2 pyruvates, 2 CoAs, acetyl CoAs, 2 carbon dioxide released. And these 4 carbons are 2 carbons containing acetyl CoA, 2 molecules, enter into the Krebs cycle, and it releases all the 4 carbons in the form of 4 carbon dioxide. So that's it. So all six carbons of the glucose is released in the form of six carbon dioxides, two at the level of no seconds no pathway, and other four in the Krebs cycle. And during this Krebs cycle, it also releases the energy in the form of capture the energy in the form of two ATPs, six NADH, two FADH2. This is another form of molecules which can be used to produce energy. So ATP, whichever produced in the glycolysis or Krebs cycle, can be utilized by the cell instantly, but the energy produced in NADH in the form of NADH in the glycolysis, NADH in the formation of acetyl-CoA, or NADH and FADH2 in glycol Krebs cycle or citric acid cycle. So all need to undergo the electron transport chain. So this molecule, through this electron transport chain, it will produce the ATPs. So all these produce equivalent to 32 or 34 ATPs through the electron transport chain, and where it needs the oxygen to capture the protons released in this process and then to produce water. So that's why oxygen is very important for us to consume. So we have to inhale the oxygen constantly, continuously, because without this oxygen, this electron transport chain cannot occur. Without electron transport chain, we cannot produce the ATP, which is needed for the basic metabolism of the body, and also basic energy needed for all the parts of the body. So oxygen is needed compulsory. So to undergo this electron transport chain and to produce the energy. And so that's why this ATP, whichever producing in the first as instantly, those called as instant ATP production, instant phosphorylation, it is called as. And this is electron transport chain phosphorylation. Okay. And thus these NADH and FADH2 acts as a check to go to the bank and get the cash. So even though you have NADH, FADH2, if you don't have the electron transport chain, you cannot convert into cash. That is the ATPs. So that is very important. So these are all steps we are going to understand next few classes. And today we will try to have a look how glucose converts into pyruvic acid. And our more interesting is how it produces the ATP and NADH. So we will try to capture which reactions are doing this in this glycolysis. So cellular level, where these reactions occurs. So glycolysis, as just discussed, it will occur in the cytosol. All the glycolysis pathway until the pyruvate, it occurs in cytosol. And instant producing the energy. This is called as substrate level phosphorylation. This instant phosphorylation called as substrate level phosphorylation, where ADP is converting into ATP during this reaction, not undergoing the electron transport chain. And this pyruvic acid need to convert into acetyl-CoA and enter into the Krebs cycle. Everything occurs in the mitochondria. And the Krebs cycle also produces the substrate level phosphorylated ATPs, and also it produces NADH, FADH2, this NADH, all these enter into the mitochondria, whichever here is need to be entered in mitochondria, which are produced in the Krebs cycle, already present in mitochondria, will undergo electron transport chain as well as oxidative phosphorylation, which produce the ATP. This phosphorylation we call as oxidative phosphorylation, where the ATP produced in the glycolysis and Krebs cycle we call as substrate level phosphorylation. And by end of these all the reactions, one glucose molecule will produce 10 NADH, 2 FADH2, and 4 ATPs. All carbons of the glucose will released as carbon dioxide molecules. So this is the first phase of the glycolysis, that is glucose convert into two triose phosphates, that mean glyceroldehyde 3 phosphate. So this illustration will gives you idea the number of carbons wise what is going on. Glucose 6 phosphate, sorry, glucose which is 6 carbon, in few reactions it will be another hexose 6 phosphate which is also 6 carbon, and this going to split into two molecules which is containing 3 carbons. After that, it in few steps it will be converting into pyruvate which is still 3 carbons. Okay, so this is for your mind mapping what is going on, and each steps we are going to see now. Okay, so this is another step. When NADH produces in this glycolysis need to undergo the electron transport chain. What do you understand just now, electron transport chain? So NADH will convert into, not NADH, NADH energy will be captured to produce the ATP molecules, and it will release back the NAD in the electron transport chain, but in presence of oxygen. So electron transport chain occurs in presence of oxygen. Oxygen need to be taken the protons released in the electron transport chain, then uh, produce the water molecules. Thus, NADH can be utilized to form the ATPs. But in case you don't have enough oxygen or without oxygen anaerobic conditions, what happens? NADH will be accumulating inside the cell, which is toxic. So we need to have a cell need to have an alternative pathway. What it will do, NADH will be again revert back giving the NAD, and it produces the lactate. Pyruvate will convert into lactate. So that we will see at the end of this glycolysis. So overall, for your understanding or mind mapping, aerobic conditions, glucose will convert into 2 pyruvate, 2 ATPs and 2 NADH will be synthesized. In anaerobic conditions, glucose convert into 2 lactates, only 2 ATP synthesized. NADH will be utilized to convert from 2 pyruvate into 2 lactates. So no NADH here, only ATP is produced in anaerobic condition. Okay, so priming stage and splitting stage and oxidoreduction phosphorylation stage. So priming stage and splitting stage you can see in the first phase, and the second phase you can see oxidoreduction and phosphorylation stage. So why we call as all these names, you can understand once you understand what is going on in each step. So in the priming stage, what happens in priming? So it is giving like investing steps here. So when you do any business, you need to have first, in order before earning, you need to invest some money, right? So that's the glucose in order to produce some energy. So you need to be prepared first. So glucose first, in order to prepare, glucose need to be primed or phosphorylated with phosphate. So glucose need to be converted into glucose 6 phosphate. Glucose converts into glucose 6 phosphate. And where the phosphate is coming from? So we are utilizing ATP. So in this step, so glucose, you can see here more clearly, glucose is converting into glucose 6 phosphate. What is going on? The sixth carbon of the glucose, so instead of OH, it is going to be like OP. That means phosphate is going to be added. From where this phosphate coming? ATP is converting to ADP, the third phosphate which was present in the ATP will be attached to this one. So become phosphorylated. So this glucose molecules once attached to the phosphate we call as glucose 6 phosphate. Because sixth carbon is attached to the phosphate, we call as glucose 6 phosphate. And during this step, this is need to be catalyzed by an enzyme. So either glucokinase or hexokinase. Any of these enzyme catalyze this reaction. And when you go into detail of this enzyme, so the hexokinase present in almost all cells of the body, and glucokinase specially present in the liver. And these reactions need magnesium ions as the cofactors. I think you have heard the enzymes and what are the enzymes and the catalysis and also cofactors, right? So for this enzyme, we need magnesium ions as a cofactor in order to produce glucose 6 phosphate from glucose. This is the one direction, so it cannot be revert back. So see the arrow, it is very important. So that means glucose 6 phosphate cannot be given back to the glucose. It is understandable, right? Once you utilize this ATP, so in order to go back, ADP to produce, it's not that easy. So glucose, once convert glucose 6 phosphate, cannot be revert back to glucose easily by this enzyme. Okay, the next one is glucose 6 phosphate converting into fructose 6 phosphate, but there is no change in the atoms wise, all are same, but only the spatial arrangement, structural arrangement is different. Here the cycle contains the 6 atoms. Here this is the pentose kind of thing, uh, the ring. Okay, so but still the sixth carbon is also there. Okay, so it contains, yeah, this is the first carbon here, second, third, fourth, five, six, but it present as a fructose. So this glucose 6 phosphate is converted into fructose 6 phosphate because they are just a special arrangements. So each can revert back to each. Glucose 6 phosphate can convert to fructose 6 phosphate, or fructose 6 phosphate can be converted into glucose 6 phosphate. This conversion um, is catalyzing by phosphohexose isomerase, because these two are isomers and easy to remember this reaction as changing the isomerism. So the enzyme called as isomerase. And the molecule here is hexose molecules because 6 carbon containing and containing the phosphate also. So the enzyme name is phosphohexose isomerase. And then this fructose 6 phosphate then again will be primed with another phosphate at the first position, first carbon, by again utilizing another ATP. And here the enzyme name is phosphofructokinase. So because this is the fructose and it is containing phosphate, phosphofructo, that is kinase. Kinase means always the enzymes which is adding the phosphate group to the molecule or the substrate we call as kinases. Here also is a kinase because it is adding phosphate to the substrate. Here also this enzyme adding phosphate to the substrate, so this is also kinase. And this enzyme also requires some magnesium ions as the cofactor, and this reaction also in a single direction. And the resulted molecule we call as fructose 1 and 6 bisphosphate. So two phosphates are there. So Latin word is bis. So fructose 1,6 bisphosphate. So because first carbon and second carbon, sorry, sixth carbon are containing the phosphates. So overall, this can be understand as a priming stage where we are investing the ATPs, okay, here to produce glucose from glucose to fructose 1,6 bisphosphate. This one fructose 1,6 bisphosphate is ready to splitting into two molecules. Okay, so this fructose 1,6 bisphosphate will be splitting down into two molecules, and those are dihydroxyacetone phosphate. Another one is glyceraldehyde 3 phosphate. When you study the chemistry, you might have seen the aldehyde structure and ketone, right? So this is the ketone. So C double bond O is the ketone. This is the dihydroxyacetone. So because two hydroxyl are there, so dihydroxy. This is the ketone acetone, and one of the place it is having the phosphate, the phosphate. So thus the name is dihydroxy. This is the first carbon and third carbon is having hydroxy, right? But of course, first this carbon is having the phosphate moiety here, so that we can spell out at the end. So dihydroxy acetone phosphate, and this one is aldehyde. COH is our aldehyde, right? So glyceraldehyde 3 phosphate. So this is whole molecule is a glyceraldehyde, and the third carbon is having the phosphate. So glyceraldehyde 3 phosphate. And these two molecules actually the same, just splitting at the middle, and basically all are same number of carbons, hydrogens, and phosphates, oxygen. So thus they are able to interchange acetaldehyde 3 phosphate to dihydroxyacetone phosphate, dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. This conversion is catalyzing by enzyme phosphose triose isomerase. Previously hexose isomerase, right? Because 6 carbon containing carbohydrates are changing isomerism. So here the isomers are 3 carbon containing, so phosphate triose isomerase here. Okay, so these two molecules whichever produced acetaldehyde 3 phosphate, sorry, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate become two glyceraldehyde 3 phosphate, because the dihydroxyacetone phosphate will convert into glyceraldehyde 3 phosphate. Okay, this is what the splitting stage. So splitting stage is one reaction, and priming stage is the 1, 2, 3 reactions. So four reactions together we can call as a first phase. This is just for your understanding. So no need to be compulsory to mention anywhere it is needed as a priming or splitting or first or second phase, just to have your understanding only. Okay. So then glyceraldehyde 3 phosphate giving rise to the pyruvate. That is what the pending things. That is called as the oxido reduction phosphorylation stage, or the second phase simply. So what happening here? Two molecules of glyceraldehyde 3 phosphates are converted into two molecules of pyruvate or lactate in case of anaerobic condition. So what happening here? You can see in this reaction. So glyceraldehyde 3 phosphate. So what is here? This is the first carbon, this is the second carbon, and this is the third carbon. Third carbon is having phosphate, glyceraldehyde 3 phosphate. The first one, it is adding it is taking back the hydrogens, and NAD is converting into NADH and H plus. And one inorganic phosphate directly added. So it is not, this phosphate is not from the ATP. So this another phosphate will be added directly from the inorganic phosphate. Okay, and this glyceraldehyde 3 phosphate must have high free energy, and which is releasing in the form of formation of NADH, and it also produces the product 1,3 bisphosphoglycerate. So this is like an acid here, So COOH, what happens like an acid, COOH, isn't it? So thus it is called as a glycerate or glyceric acid. And bisphospho, because two phosphates are there at the position 1 and 3. This is the first carbon and third carbon, both are having phosphate. So thus it is called as 1,3-bisphosphoglycerate, and this reaction catalyzing by glyceraldehyde 3-phosphate dehydrogenase. So what is happening here? Two hydrogens are taken and released or giving to the NAD molecule. So thus dehydrogenation is happening. So dehydrogenation, the reaction and catalyzing by the enzyme dehydrogenase. So the substrate is glyceraldehyde 3-phosphate. So when the glyceraldehyde 3-phosphate is converting into 1,3-bisphosphoglycerate and dehydrogenation is happening in this reaction, and the enzyme catalyzing by the dehydrogenase and the substrate as glyceraldehyde 3-phosphate, the enzyme is glyceraldehyde 3-phosphate dehydrogenase. And during this step, you remember NADH is producing and inorganic phosphate is utilizing. Okay, so this 1,3-bisphosphoglycerate, what is going to be happen next? So the phosphate which is added to the first carbon is going to be released, and released how? It will be producing the ATP. So ADP containing 2-phosphate, right? Adenosine diphosphate, right? So it need to be converted into triphosphate. One phosphate need to be given. So from where it is going to come? From this molecule. So this phosphate to use to produce the ATP from ADP. So it's not only the phosphate giving the energy, free energy from 1,3-bisphosphate glycerate is giving to produce the ATP from ADP, and the product is 3-phosphoglycerate. So this is only the third carbon is having the phosphate now, and this is still acid because already the hydrogens removed, right? So because of that, it become acid here now, and this is 3-phosphoglycerate. The enzyme catalyzing this reaction is phosphoglycerate kinase. Okay, so this is actually, interestingly, the bidirectional. So that means 1,3-bisphosphoglycerate can convert into 3-phosphoglycerate, or 3-phosphoglycerate convert into 1,3-bisphosphoglycerate. But in the reverse reaction, ATP need to be utilized. It is not going to be spontaneous. Okay. And in both reactions, phosphoglycerate kinase is the enzyme catalyzing. So if you take the reverse reaction, the substrate is phosphoglycerate, right? And what is going to be happen? Phosphate is going to be added. So thus it is kinase. So for the reverse reaction, based on that, the enzyme name was given phosphoglycerate. This is the one, and kinase, adding the phosphate. And for this enzyme also, magnesium is a substrate cofactor, and ATP is synthesizing. So energy producing steps are very important to remember. So here NADH produced and here ATP produced. So next is what? Just a special arrangement. So third place phosphate is moving to the second place. So this OH is going to third place. That's it. So that's 3-phosphoglycerate becoming 2-phosphoglycerate. The enzyme needed for is phosphoglycerate mutase only. Okay, the next one is water molecule is going to be deducted from this molecule. So released water molecule. How to release? You can see this OH from the third carbon and hydrogen from the second carbon will combine and release as a water. So each carbon need to be have how many bonds? Should have the 4 bonds, right? So what happens if you remove this one hydrogen? This carbon will be having only 3 bonds, right? So if you remove this, another 1 bond will be pending. What happens if you remove the hydroxyl group from this CH2 OH? So this carbon also will be having only three. One to this carbon, another to hydrogen and hydrogen, and another supposed to be hydroxyl. But if you remove the hydroxyl, this carbon also have one bond, scarcity. So what happens? This carbon and this carbon will have double bond for a third and second and third. So that is going to be resulted. So once you remove the water molecule from the 2-phosphoglycerate, it will be phosphoenol pyruvate. So what is mean by enol? You remember the chemistry. Enes mean alkenes, the double bond ones, and all mean this alcohols, hydroxyl groups. So here the double bond is there, all alcohol OH group is there, and acid also there. So phosphoenol pyruvate, pyruvic acid. So three carbon containing acid. This is pyruvic acid. So phosphoenol pyruvate. This is the molecule. It will result. So the enzyme catalyzing the dehydration. This reaction is dehydration, isn't it? Water molecule is removing. So this dehydration uh, reaction catalyzing by enolase enzyme. Then this phosphoenol pyruvate is converted into pyruvate. It's at the beginning it is called as enol pyruvate or pyruvate. What is the thing? So what is happening here? This phosphate need to be released and added to the ADP to produce the ATP. So ATP is synthesizing in this step. So that means free energy of the phosphoenol pyruvate is actually giving some of it to the formation of ATP and remaining to the pyruvate. And here magnesium ions are the cofactor for this enzyme pyruvate kinase. So this is actually the the phosphate is releasing, but it at the beginning it was named when it's not clearly understand. So it is still we are using the traditional name pyruvate kinase. Okay, so what is happening? Just a phosphate is releasing, and hydroxyl is will be kept here. So here the ene group also there, and all also there. So enol. This is the pyruvate, but enol structure. But spontaneously it will converted into this keto group. Okay, so this double bond moves to here, and hydrogen moves to here. So O will be having the double bond with C second carbon, and this methyl group become a CH3. Okay, so this is the also pyruvate. The name is pyruvate, but it is called as keto form. Okay, this pyruvate is the end product of the glycolysis. Okay, so what is happening here? The how where are the energy is released? So NADH released from glyceraldehyde 3 phosphate to 1,3 bis phosphoglycerate, but one molecule of glyceraldehyde 3 phosphate only we are seeing this reaction. So another glyceraldehyde 3 phosphate also undergo the same reactions and produce the pyruvate. So that from one glucose molecules, two NADH are going to be produced. One glucose molecule, and here this step 2 ATPs, and this step 2 ATPs are going to be produced. So totally how many ATPs are produced? This here 2 and this here 2. 4. And the previously priming step is only the one glucose, there is not two cycles. So priming step only here one ATP and here one ATP, two utilized. So from one glucose, it produces two pyruvates, two ATP produced but two ATPs utilized. So net yield is two ATPs and two NADH. Okay, so when you enter the NADH into the electron transport chain, it will synthesize equivalent to three ATPs. NADH will produce equivalent to 3 ATPs. So conversion of glucose to 2-pyruvate, 4 ATP synthesized, respiratory chain linked oxidation, that means electron transport chain, to NADH, we can write as 6 ATPs because each NADH will produce equivalent to 3 ATPs. So that's 6 ATPs we can consider. So one glucose molecule produce 10 ATPs to produce 2 pyruvate molecules. But we are utilizing in the priming stage 2 ATPs. So 8 is the net yield. So primary yield is 10, but net yield we have to say that only 8 ATPs will be produced in glycolysis in aerobic conditions when glucose convert into 2-pyruvate. What happens to the anaerobic condition? This is we need to understand. So when anaerobic condition, that means, so until this pyruvate is still it is still continue until here there is no change. That means glucose molecule will produce the pyruvate, 2 pyruvates, and ATP is produced, ATP is utilizing in anaerobic conditions. In anaerobic conditions also, in this step, glyceraldehyde 3-phosphate to 1,3-bis phosphoglycerate, NADH produced. Everything is the same. What happens? The cell is under the anaerobic condition. That means NADH cannot undergo the electron transport chain. That is, you need to understand. So electron transport chain only can convert or utilize the NADH to produce the ATP. Otherwise, NADH is just keep accumulating there because oxygen not there. It cannot undergo the electron transport chain. Thus, there is no converting the energy into ATPs. So, so you can see the solid line here. That means in aerobic conditions, NADH will enter into the respiratory chain and produces the 3 ATPs and give back the NAD which is needed for the glycolysis pathway. But in anaerobic conditions, so NADH is keep accumulating, so which is not good for the cell. And moreover, NAD will be not available anymore. So when many glucose molecules are there, want to undergo glycolysis to produce the energy even though the anaerobic condition, but when there is no NADs are available, it cannot proceed these steps where the ATPs are produced in these steps, right? So when there is no ATP, it is fatal for the cell. It cannot survive. So cell need to go and undergo an alternative path. That is, NADH need to be utilized just to replace the NAD first of all. And NADH accumulation poison is going to be toxic, also need to be reduced. So thus NADH utilized to convert pyruvate into lactate. So what is happening here? The dehydrogenation or hydrogenation is happening. So this hydrogen molecule atoms of the NADH is added to here to the second carbon. So double bond O become OH and H. So this is going to be another OH. Okay. So thus the pyruvate is converted into the lactate or lactic acid. Still it is acid, right? So you can call as lactate or lactic acid. Here also pyruvate or pyruvic acid, both are same. And utilizing the NADH here and releasing the NAD there. This NAD now available to continue the another glycolysis pathway. So glycolysis pathway should not be stopped. It need to be continuously occur. So when you not have enough NAD, glycolysis is going to be stopped. So it should not, it should not be get resulted. So thus, so by undergoing the anaerobic pathway, the pyruvate will convert into lactate and NAD will be given back for the next cycles of glycolysis. Okay, so Because of that, NADH is utilized, so we cannot take that one as consideration in producing energy. This 6 we need to be removed. So what is the net? 4 ATP synthesized and 2 are utilized. 4 minus 2, the net yield is only going to be 2 ATPs will be produced in anaerobic glycolysis from glucose to 2 lactates. So this is the cycle, the all the 10 steps of the glycolysis, where it will glucose undergo either anaerobic or aerobic splitting of the glucose into lactate or pyruvate respectively, and will produce 2 or 8 ATPs net ATPs production respectively. And then we will see the importance of this glucose in